Hi, welcome to Chapter 10. This is photosynthesis. I'm going to be drawing some figures from several different places here, so they may not line up exactly with your textbook. So what photosynthesis does is takes the very low energy molecules, CO2 and water, and can break them apart, which is no small feat, and rearrange the atoms and the electrons into a sugar, some water, and waste product oxygen. Photosynthesis, as the name implies, is broken up into two parts: the photo part, called the light reactions, and the synthesis part, where we're going to make sugar, and that's the Calvin cycle. So the light reactions occur in the grana on the thylakoid membrane, and their job is to capture the energy from sunlight to split water, releasing the electrons and the hydrogen ions, which will shuffle the electrons around. We're pulling those hydrogens off, so we're left with oxygen, which the plant will release as waste product. We're going to make ATP and NADPH, which is very similar to what we learned in cell respiration. The Calvin cycle occurs in the stroma, the thick fluid outside of the grana, or the thylakoid membrane. And here we're going to take that energy that we just stored in ATP and NADPH, and we're going to be able to donate those electrons and that energy to carbon to form simple sugars. So here's an overview, a picture of what it looks like. There you can see the thylakoid membrane is receiving the water and the sunlight, producing ATP and NADPH, which then goes over to the Calvin cycle, where we're going to get a simple sugar, and that's in the stroma. So the key to the first part, the light reactions, is capturing light. And the real molecule here that does this is chlorophyll, specifically chlorophyll A. It's going to receive a majority of light in wavelengths of around 700 nanometers. And when chlorophyll gets struck by light, the electrons in the chlorophyll are going to get excited, and you're going to be able to capture that excited electron to harvest the energy out of it. So that's sort of the magic of chlorophyll. So the photosystem is arranged in, like this picture shows, it's a bunch of chlorophyll molecules, up to 300, as well as chlorophyll B and carotenoids, some other pigments. So you have this light harvesting complex called a photosystem. There's going to be two of these. The first one is photosystem 2, because it was discovered second. You're going to be able to capture light, pass that energy from the light down a chain of pigments, and basically release that electron into the reaction center. So we're using the light to capture electrons. The pigments are bound to the reaction center protein molecules, and again, they're going to be able to funnel that energy from the light into the reaction center to sort of concentrate the energy. A single electron gets basically bumped up to a very high energy state, and there's a molecule there called a primary electron acceptor that will receive it. Again, this is located on the thylakoid membrane, the inner membrane of the chloroplast. And we're going to go through a process now with that electron called non-cyclic or linear electron flow. So we have an excited electron, and it's going to be transmitted down an electron transport chain. Now, if you remember from cellular respiration, an electron transport chain's job is two things. It lowers the energy level in the electron, and as it's doing that, it's pumping hydrogen ions across a membrane, creating a high hydrogen ion concentration. Just like in cellular respiration, that high hydrogen ion concentration will flow back through the protein molecule ATP synthase and create a bunch of ATP. It's a spinning turbine. Where this is different now than cellular respiration is that electron doesn't fall all the way down in energy. The electron gets received by another photosystem, photosystem 1. That photosystem will harvest more light energy and re-elevate the electron to an even higher level. So to trace the electron, we got elevated in photosystem 2. Some of that energy was used in the electron transport chain to create ATP. The electron then gets bumped up again, and now with that very excited electron, it has enough energy to join with NADP+, the battery, and store that electron's energy in NADPH. So we've just made a bunch of ATP, and now we're making NADPH. So we've gotten ATP in batteries using just water and light energy. Here everything is together on a thylakoid membrane. The light comes in, photosystem 2, electron gets elevated, electron transport chain spins a turbine to make ATP at the bottom there. The electron gets re-elevated, which now has enough energy to produce NADPH. So what we've just done is stored a bunch of energy. Now, under some conditions, that linear flow that we just went through, the electron followed a linear path, doesn't create enough ATP. So what we get is a surplus of batteries and not enough ATP. So in the cyclic flow, the batteries that we just created go back and donate their electron to the electron transport chain, which then is going to continue to run the mill, the ATP synthase mill. So that's the cyclic part. We've created our batteries. We have too many batteries, so some of the electrons get redonated. The electron falls down the electron transport chain, creating more ATP. The electron then gets re-elevated, and we charge our battery all over again. So that's the cyclic part. And as you can see in this diagram, photosystem 2 isn't even used in this because we're using the electrons from the battery instead of the electrons from water. So now we have a bunch of energy stored in the form of ATP and NADPH. Now we go into the Calvin cycle, and this is very similar to the cellular respiration process, the citric acid cycle. If you recall, the citric acid cycle is going to use up our sugar and release CO2. Now we're just doing the reverse. We're taking CO2 out of the air, we're adding the energy to it, and we're going to make a sugar. So it's essentially the reverse reaction. And all of this is going to take place in the stroma, the thick fluid outside of the thylakoid. So there's 3 phases. Carbon fixation. The first and hardest part is getting the CO2 out of the air and broken apart. So that's our first step. Then we're going to reduce it. We're going to gain electrons. That's reduction. Those electrons are going to come from our batteries and our ATP. Once we drain that energy out, we're going to get a little bit of stored carbon, but now we need to regenerate the process. And that's the third phase. We need to be able to accept CO2 once again. So here everything is. You can see the carbons, CO2s, enter. There's an enzyme rubisco, which is able to grab that CO2, and along with a molecule of RUBP, the rubisco is going to be able to take a CO2 and stick the CO2 onto RUBP. Once we get that, that's the carbon fixation stage, we can put a bunch of energy into that that molecule we just made. So we're going to dump 6 ATPs, 6 NADPHs into that molecule. And from it, we're going to be able to remove 1 G3P sugar. So that's the equivalent of a pyruvate, a 3-carbon sugar. Now that we have our sugar, we need to reset the cycle. With that, it's going to take more ATPs to go back to the original RUBP molecule. So for this process to make a single G3P sugar, we're using 9 ATPs and 6 NADPHs. That's what we just stored in the light reactions. As a side note, Rubisco's terrible at doing this. It's really, really inefficient, but because all the energy that we're storing is free, it's good enough. Plants can work with that. Under certain conditions, though, Rubisco's so bad that it actually can harm the plant, because it'll incorporate oxygen instead of CO2, and if you run oxygen through the Calvin cycle, you get no desirable products, and what you get out is actually harmful products. So Rubisco is not an ideal molecule to use here, but it's what the plants have. In certain plants, C4 plants like grasses, bamboo, sugarcane, they recognize that Rubisco is not great. So what they do is they separate out the light reactions and the Calvin cycle into different cell types. The light reactions are going to occur in your normal mesophyll cells, but a lot of the ATP and other things are going to be shipped off to a specialized cell called a bundle sheath. And this is where the Calvin cycle is going to go. It's going to happen. So you have 2 different cell types sort of dividing up the work to limit the amount of negative products that we're going to get from the Calvin cycle. Another type of photosynthesis, called CAM on the right there, goes even further to separate out the light reactions in the Calvin cycle by night and day. They also are going to have bundle sheath cells, but they just don't run the Calvin cycle except at night, when their stoma can be open, accepting CO2, and they don't have to fear any extra water loss out of their stoma. So, to summarize, light reactions are going to harvest light and split apart water, really cool, make ATP and NADPH out of that. You're going to take that energy, be able to grab CO2 out of the air, and get a single G3P molecule as a result.